Welcome to our Micro JC Summer Book Club discussion. This is our second book club for July. And this time, for this month, keeping with our theme on evolution, we're going to be talking about this book, which is A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution by Jennifer Doudna and Samuel Sternberg. This is a book that's basically about CRISPR, the CRISPR technology, and then the implications for CRISPR technology for evolution, and giving us some power, essentially, over evolution and some ability to kind of direct evolution of humans and other animals and plants and so forth. There's a lot in here. It's a really well-written book, though. It was a nice read for the public, I think, to try to understand this new technology and what it means. But before we launch into it, we'll do a little bit of introductions. So I'm Laura Williams. I'm an assistant professor in the biology department at Providence College in Providence, Rhode Island. And next to me, we've got Nicole. So Nicole, do you want to introduce yourself?
Tweet is old dusty on, and I work at the University of Northern British Columbia, but I will soon be joining in Montreal, where I'm currently right now looking for apartments. Looking forward to it because a great example of medicine is a really great way to bring non-scientific experts a clean narrative on understanding gene editing technology.
We're losing Nicole a little bit. Nicole might kind of come in and out.
That are really strong pieces of Jennifer Doudna and Sam Sternberg story.
Well, I think we're having trouble. So Nicole is coming to us from Montreal, which is not her normal place that she's going to be. So one of the things that might happen is that Nicole might be bouncing in and out a little bit. So she is unfortunately on hotel Wi-Fi, which makes for a little bit of a choppy kind of interaction here. So we'll do our best because Nicole has made a good effort to try to be here for this, even though she's traveling. So she's actually, I think what she said was that she's looking for apartments in Montreal because she's about to make a job transition, which is very exciting. And so we'll try to like muddle through with the Wi-Fi and see how that goes. Meanwhile, we've also got Lucy with us for the first time. So Lucy, do you want to introduce yourself?
Sure. Hi, I'm Lucy Mccully. I'm at Micro Mccully on Twitter. Mccully is MCCULLY. I am a PhD candidate at the University of Massachusetts in Dartmouth. So we're on the south coast of Massachusetts. And I study soil bacteria and look at interactions between bugs. And I'm very excited for this. It was a very interesting book.
Yeah, I was pretty excited about this too. I thought it was a nice, I was really impressed that I felt like it made what could be an extremely complex topic really accessible to the public. And so it's funny because just an hour ago, maybe, I think, so my not to like get too personal with stories, but my mom is actually currently getting chemotherapy for cancer. And she is at her chemotherapy appointment right now. And she texted me and said, I'm looking at the New York Times, and there's a story in the New York Times about this new gene editing technique that could be really important for cancer treatment. I was like, yeah, we're literally just about to talk about a book about that. So and Jennifer Doudna and Samuel Sternberg, in writing this book, do a really good job of kind of making it clear the vast implications that this technique has for medicine, for agriculture, for research, for, take your pick, and you can find a way that CRISPR is potentially going to have an impact on that particular area. So I would really, anybody who's watching this, I would really strongly encourage people to basically, if you want to learn a little bit, if you've heard this term and you're like, I need to figure out what's going on with CRISPR, or maybe you want to teach it in class. I think this is an excellent resource for teaching this to students. The book is laid out in a really accessible way. It starts out with a description of kind of the history of how CRISPR was discovered and how the mechanism behind CRISPR was kind of teased out. And then she starts going into kind of applications of CRISPR, and then wraps in kind of some of the ethical and other issues that surround how we deploy this technology and what we choose to do with it. So before we kind of get, I think that breaks it up really well, but before we kind of launch into discussing particular parts of the book, I wanted to actually see what people's overall impressions were. I see that Nicole is on the move. I think she might be trying to see if she can get to a better access point, but until she gets settled, Lucy, do you have some overall thoughts about the book?
Yeah, I thought it was a very interesting read. It was very approachable, I think. I got the impression that maybe not anybody, but almost anybody could pick up the book and actually like follow, this is how we worked this out, this is how we got to understand this. And that was also really fun for me. Actually, I mean, I knew about CRISPR as the immune system for bacteria, but I hadn't actually learned about some of its applications. That was very cool for me to learn about. What I found it was very heavy on was sort of the bioethics, and that was maybe harder for me to read about, only because I don't have a strong training in philosophy and that kind of thing. So it was very, very different for me to have to start to think about absolutely all of the different implications that using this technology can have, which is not a small amount.
Yeah, I agree, and I think she's one of the strategies that they use in this book is that she. There are two authors, and the second author is Jennifer Doudna is a name that I think people who know about CRISPR would definitely recognize as being associated with the very early stages of studying this mechanism. And then Samuel Sternberg is a student of hers, a former student of hers, who now runs his own research lab. And the way even though they co-wrote the book together, the way they chose to present it was in her voice only, kind of as a narrative story from her perspective. And I think that was useful because I think she makes it very clear when she's talking, to your points about bioethics, she makes it very clear when she's talking about this, that was a major shift for her as well. That originally she kind of approached it from a standpoint of, she was an RNA biologist, and she was approached by a colleague, Jill Banfield. I was like, her name is escaping me. I was like, approached by a colleague, and that was her inroad into understanding the system. And the more that they got into it, and the more they saw it kind of accelerate, the more she realized that there were some very far-reaching implications. And she kind of calls back to recombinant DNA technology in the 70s and the meetings that took place around that, which we had a previous book club summer about genomics, and there was one of the books we read talked at length about those meetings and how they were set out to try to kind of lay some groundwork for, what are our responsibilities as a scientific community? What kind of guidelines should we have? So I think Lucy, I think your thoughts about bioethics, I think she would probably share them as well. Like, I don't think that she felt very comfortable getting into that either. It felt like she should. Nicole, are you resettled? Do you wanna give it a try and see if we can keep you?
Yeah. So, am I articulating coherent sentences at this point?
Yeah.
I'll try to be okay.
That sounds great.
Okay, fantastic. So I actually really appreciate that Dr. Doudna stepped into talking about having to learn policy outside of her silo, and I think that's where all of us has started with academics. Is we're trained way, for like CRISPR editing and all gene editing in all its manifestations that were covered in the book. That broader implication of what we should be modifying, what the human so-called born with or emergent biological properties from life cycle versus us doing things to them, possibly to make things better. If you're on the edge of technologies, or if you're on the edge of sciences that are addressing that directly, you can't not think about it as a member of society, which even as academics, we all inevitably are. So it's very important to see that. I think she wrote as honestly as she could have based on where she was at the time. And I also like the fact that she acknowledged that her values have been shifting, you know, in the process of trying to understand the broader implications of the work. But also from a teaching perspective, I think that part of the book is really good for discussions with class of the work, but also just the whole package of how she organized talking about the game plan of why you want to modify a genome, again, using the narrative of disease, getting into genetic modification of crops, but also using some really nice visual models to go with very, very accessible writing. Scarcely word endonuclease, we heard about precise scissors, and I mean, keeping it their way to even give you the tools to re-articulate this to a classroom. And whoever did the drawings, I didn't look at the illustration credits, all arted extremely well. So get to the ground rules of things like USDA's definition of GMO and what we should be thinking about, or what the opposing viewpoints are excellent in this book. So I will mute now.
Yeah, yeah, we got probably, I'd say, like 85% of that. Only 15% was edited by your Wi-Fi. But yes, I think your major points came through. Yeah, that you did mention the drawings. There's beautiful drawings in this book. I also did not look up the illustrator credit. Maybe I can manage to do that before we close out. But yeah, I to kind of think about it a little bit from walking through the order of the book, because I think it's presented in a very logical fashion. So first, there is essentially, you know, where did this all get started? And one of the things that I think is extraordinary about this, that I think she talks about a little bit but doesn't go on and on about, is the fact that this is honestly, I think, I mean, it's not any longer than 10 years. I mean, we might be talking 5 years that this is a system that's been understood to the extent that it can be used in research labs. But this is quick. So she starts out by kind of presenting a bit of just laying the foundation to remind people or introduce people to the idea that editing the genome or manipulating genetics as a therapy or as a research technique was not new with CRISPR. That there were other techniques that were being done previously, and CRISPR basically just kind of was a rocket ship that blasted past a lot of the problems they were having with some of those, but it wasn't a foreign concept to people. And she mentions talons, and she mentioned, I appreciate, because I didn't know this, I appreciate that she made a distinction between gene therapy as a treatment and then gene editing as a treatment. The idea being that gene therapy is you have a defective gene, so you're going to supply a quote unquote healthy copy almost as a complementation, like you would do in a mutant experiment. You've got a mutated gene, then you can complement it and you can rescue the phenotype. And so she points out that that's gene therapy, whereas gene editing actually goes into the genome and says that's a defective gene, we're actually going to fix it, and we're going to see if we can restore a healthy copy from what's there already. And she kind of walks through all the various things that people have tried for that, and the challenges and techniques that they had to get over and that still remained when CRISPR kind of started to come on the scene. And that's basically the first chapter, is kind of the quest for a cure. I felt like this was really accessible. I don't know if you guys had any thoughts about kind of this introductory groundwork laying chapter where we kind of go over where were we before CRISPR came into things. Any thoughts on that? Yes, Nicole, go for it.
I really like the cohesion of that first chapter, because when I first took molecular biotechnology in undergrad, my textbook was one where you learn the parts of shuttle systems or the parts of beacon probes. And I mean, trying to actually give the ground rules for why the pathological conditions that she discussed were the pathological conditions that they were, and the implications of what the type of mutation were, whether it was point, whether it was a frame shift, even though she didn't use those words. The transmissibility, so the whole thing with choke virus and noticing that arginase level in humans, so getting that very plain language description of how transferable shuttling systems for a complementation could be worked really, really well in this chapter. I think that it's very clean writing, and I also thought that it was brilliant that this roadmap of different strategies was actually mirrored when she started talking about the locations, basically, of gene editing for somatic versus germline therapies later on. So there was clearly a lot of care put into setting up the text and the mechanics of how this was going to work to keep the brains of the reader thinking along the same sort of stepwise progressions. And I really, really appreciated that.
Lucy, do you have thoughts on this kind of first chapter?
I did like, I think basically what's already been said is I like the groundwork presented here, and I do think it would make a nice like background for anybody maybe learning about this for the first time, to kind of, it contextualizes it. I think it's a nice, and this must have also been, I imagine, challenging for the authors, because I imagine a lot of this was out of their wheelhouse, right? But so that they can talk about where CRISPR is now, you first have to introduce all these previous things that they don't necessarily think about on a day-to-day, which I thought that was really, really neat and very helpful.
Yeah, I can imagine this is a lot like, I wonder how difficult it is, because this whole chapter is like a review, but it's a review that you can't write like an academic literature review. So I wonder how hard it was to shift out of, if they found themselves starting to write like academic lit review type style and have to be like, no, we can't do that, because this really is just the tip of a very large iceberg, that they had to kind of think about, what are our overall goals, and we're trying to get the reader from point A to point B, and we don't need to go off into like A sub 7. And kind of like we have to like keep moving forward. I think they did that well. So after that kind of groundwork, laying that groundwork, then she gets into kind of in the second chapter, her introduction to CRISPR, which she presents as coming to her from a colleague at University of California, Berkeley, Jill Banfield, who is a microbiologist, a very eminent microbiologist, and who had kind of come across the system and was looking for an RNA biologist to kind of help her make some sense of it. And so that's kind of how she presents her introduction to it. And then she tells the story of kind of what's going on with CRISPR from a narrative perspective, like how it progressed. She makes mention of a number of different people who kind of had contributed. She talks a little bit about, one of my favorite details when I've taught CRISPR in my genomics class before, is that I think that the first person to have published about this, I can't remember his name now, unfortunately, because names are currently escaping me very easily. But I think the original paper where the person kind of said, hey, isn't this funny, got rejected from like 16 journals or something ridiculous like that, and just ended up in some very far-flung kind of journal. And now it's like this ridiculous, like impressive technology. So that's always funny to me. I always tell the students that. I'm like, this can happen. And then the other detail that I appreciate is the mention of Danisco, which is basically the yogurt and dairy people who were trying really hard to stop their cultures from getting contaminated with phage. And so they were like, we're really tired of losing lots of our cultures to these stupid phage. How can we get these phage to stop killing our dairy-producing, lovely, fermenting bacteria? And then CRISPR. You're like, okay. And I think, I forgot to look this up, but I think there is a pizza cheese that is now made because of a CRISPR engineered lactic acid fermenting bacterium of some kind. So that's fun. Okay, one of the things that I was curious about for you guys is, I'm definitely not a CRISPR expert, but I know that it is very complicated, that it's not just one CRISPR system. In MicroJC, we've actually done a CRISPR paper before, and Joel, who was joined us at the time, had this little whiteboard where he kind of like worked out how everything worked, and it was all very elaborate. And so it's certainly not the case that if you describe one CRISPR system, that's the only way it works. And I feel like she did a good job kind of getting across what's happening without sinking too much into the quicksand of the details. So in these chapter 2 and in chapter 3, she kind of explains how it actually works and how they teased it out. Did you guys feel like you had a good sense of what was going on once you were done with those chapters?
So I ended up reading it twice, and if you asked me to describe it precisely to you right now, I would probably cry a little bit. But I mean, the difference between type 1 and type 2, and why type 2 ended up being the go-to, I guess, for editing was very clear to me. Dealing with describing any system that has a whackload of, maybe I'm going to use the term accessory proteins wrongly here, but I mean, there's a lot of help for the main event in this particular system, not unlike the complement system in immunology and other things. And I think they did a really good job of really focusing on what we colloquially always call the business ends of the system. Who's the GPS, that CRISPR RNA molecule, understanding that there had to be a second RNA molecule that they had to chuck in at the end. It wasn't just the primary recognition site in the scissors. You actually needed a bunch of other stuff. So, and explaining why they cared about sticking them together if you wanted to use this as a tool. So justifying some pieces of the engineering that came in. I also thought, again, the whole book is very clearly written. And I think when they chose to hone in on the details, it was a very judicious example. So yes, I do agree that their description of the systems was adequate for the non-science looking public and also new science looking people to CRISPR.
Sorry. Yeah, I agree. I thought there were some very clear chapters about how they got there and explaining what CRISPR would look like in the natural world of the viruses infecting bacteria and so on. Actually, I think I ended up going back through, it would be chapter 3 again, just to single out some more of those details about how they worked out the different CRISPR things. I hadn't really read that much about CRISPR before this, except for, you know, it being a bacterial thing. And that was one of the things that I had to reread for, was the justification of using this molecule over this other molecule that they'd originally looked at, because I think it was a little bit more complex, ended up using more proteins, if I remember correctly. And then also the thing where they fuse the RNA molecule, so it's a single RNA molecule ends up being a much simpler system, and how that is then downstream used in a lot of these applications. And I did think that they worked through all the details with enough, with enough detail so that you can actually follow through.
Yeah, I agree. I think it was interesting, and it makes sense to me, but I think it was interesting that they kind of, they definitely discussed, and Lucius mentioned this, and I think Nicole too, that it was discovered as kind of a, the term people like to use is that it's a bacterial adaptive immune system, which we definitely don't really think of microbes as having an immune system, let alone an adaptive immune system. And so that was the initial interest that people had in discovering this was, oh, hey, this is really interesting. We can see that there's evidence in the genome of bacteria that they're kind of collecting mugshots of these viruses that they've been exposed to. And then there's this whole protein and RNA-based system that uses that information as a defense. And the thing that I think was interesting is that they kind of, very understandably, kind of skipped completely over about like how do the mugshots get collected and what happens, and in bacteria, how does this work? And I support that, because they were heading for what are the pieces of the system that you can take out of the context of bacteria and then use as a tool. And those really are the enzyme, the Cas9 enzyme, and then the guide RNA, which is the GPS system that targets the cutting enzyme to a particular location in the genome. So they kind of dispensed with a lot of the other proteins that have to do with grabbing sequences from invading phage and kind of integrating them into it. And I think that's completely reasonable, because as a technological tool, that's not the parts that you kind of care about. You care about the enzyme that does the cutting, and you care about the piece that tells it where to go cut. And like, that's where you are. And they worked through kind of the various things that they did to kind of tweak that system a little bit, because I think Lucy was mentioning this. In the natural system, I think there's 2 RNAs that get incorporated into the final complex, and what they managed to do was make those 2 RNAs into 1 RNA molecule, just to make it simpler. So the tracer RNA, which I think has a structural purpose, is kind of combined with the guide RNA into 1 RNA molecule that sits with the protein and then kind of helps target it and so forth. So they kind of walk you through that. And then they kind of start to, in the 4th chapter, they start to kind of get into, okay, you know, how would this work if you introduced these pieces, this enzyme and these RNA molecules, into systems and kind of engineered these pieces to be able to start editing the genome in things that are not bacteria. And there's some discussion about, I liked this because it starts to walk through, in chapter 4, it starts to walk through the various ways you can edit genomes. And this is a bit that we discuss in genomics, where it's essentially, if you just have a guide RNA and the enzyme, then you're going to cut a targeted piece of DNA in the genome. You're going to introduce a double-strand break. And then, because of error-prone repair, you're probably going to introduce some mutations, and you're probably going to get a gene knockout. So that's like, if you want to use simple, that's like the most straightforward way to do gene editing with the system, is you just basically target it to a section of the genome that makes a cut. The cell's own repair systems probably make some mistakes, and then you've got a gene knockout there. And it's funny because she was talking about this, and she was talking about how people use this in research, and I was actually thinking, oh, hey, wait a minute. Because in our research in my lab, we're kind of, in the not too distant future, we're probably going to need to do some gene knockouts. And I had been thinking to myself, because we work with predatory bacteria, like, how on earth are we going to do this? People have done it, but it's like really complicated. And I was like, oh, wait, you could just do this. Oh yeah. And it's funny to me because I've been reading about Crispr, but I'd never thought that like I could use it. And she's like, oh, and people use it all the time now to make knockouts in bacteria. There you go, that's what I should do. So there's that idea. And then she kind of graduates the reader from that to the idea of adding a third part. So you've got enzyme, you've got targeting RNA, and now if you want to maybe do a replacement instead of just a knockout of a gene, you can provide a piece of template, and then the cell will actually recognize that piece of template kind of matches the ends that have been cut, and you'll get repair by homologous recombination, which should then replace any defective sequence with the wild type sequence, which is actually a good way to replace a defective gene. So if you have a gene with a mutation that's causing a bad protein product, or it's an other, Huntington's disease, you've got too many of those triplet repeats. So if you want to try to see if you can bring down the number of triplet repeats, you supply a template that's basically like, yes, this is a fewer number. So you replace the piece that you've got now with this piece, and then we'll see what happens. I felt like that made sense, but I've taught it before. So I didn't know for you guys, what did you think about kind of walking through, because once she gets done with that, we kind of launch into how does this get applied? So the knockouts and then the homologous recombination is kind of an important setup that she needs to introduce before we move on to applications. Did you guys think that that was well explained?
Yeah, and I also thought that the pictures, again, I need that sort of thing. So it was super helpful. It's interesting to hear you talk about like the possibilities for your own system, because I came out of a protein engineering background. So for me, it's never really natural to think about, we'll just make the cell make the mistakes to knock the thing out. It's always like, what are we deliberately going to put in place here? Because there's always like something, like if we're going to vandalize it, we're going to know exactly how we're going to vandalize it. But I mean, it's interesting to think about this now based on things that I wanted to do, well, wanted to know about the system that I'd done my PhD on, because I had some enzymes that came out of Pseudomonas aeruginosa. They had multiple versions of this metalloisomerase. And I always wondered, like, wouldn't it have just been great to knock out all three of them and see what this cell does with methyl glyoxyl toxicity? Because we didn't really know why it had 3 different glyoxylase 1 enzymes, let alone the fact that they belong to 2 different metal activation classes. So somewhere we'll do that one day, I'm sure, and then find out that Pseudomonas aeruginosa doesn't care, because it seems like that's how these sorts of experiments always end. They just find a way to survive. But I really did appreciate sort of the walkthrough on the techniques. And also, I really like that she brought in the gene inversion part too, because I think one thing that, especially if you don't talk about, you know, gene editing technologies or recombinant DNA, it's easy to think that there's a rule book for how you deliberately mess up a system to achieve non-functionality. And I think it's good that she actually went through a little bit of that, because, so that you understand that, like, there's different ways that, you know, DNA can rearrange itself to sort of achieve knockout.
Yeah, that was also a thought that I had while reading through this, was that I could also use CRISPR. Because we do, in my lab, actually do knockouts pretty regularly, because our system is fairly easy for that. But I was like, but wait, you could do it even faster, and it would work really well. So now I have a hankering to use that and see how well it works, because part of our system is we have like all the tools for it and stuff, but we have another bug that we have absolutely no tools for. So that would be very, very nice if we could use that. This is also the chapter where they start talking about putting the system into like mammalian cells and animals. And I did like the explanation for that, because I very rarely work, or have only like once worked with mammalian cells. And so that they actually explain, you know, you have to get it into the nucleus, and here's the ways that you can do that. And I thought it was very approachable, and, you know, it made me remember that, oh, that is a thing that you have to do. So I liked that. And then, you know, I think then they started to explain how they started putting it into animals and plants. And so I'll let you introduce that part.
Yeah, that's an excellent point, because I think, so the first part, which really walks through the mechanism and how it's all done, is about 115 pages. So it's a little less than half the book. But I think it's really important, because the second half of the book, just like Lucy mentioned, is kind of gets into applications, and then you have to start asking questions about ethical guidelines and how should we be shaping how this technology is deployed. And if you don't really understand what it's doing, it's hard to really get a grip on how it's being used, and how she draws a lot of important parallels to existing technology, especially when she gets into like human reproduction. That she draws a lot of parallels to currently used technologies and how those are or are not regulated. And so to understand the policy parallels, it's important to kind of understand what this technology is doing. And so, yeah, Lucy, you're absolutely right. Like, it's not really just as simple as like you throw some protein and some RNA at some cells and then everything's fine. It's becoming really accessible to lots of research labs, but there are specific things that have to happen. Otherwise, it just seems like it's like, you know, Frankenstein. You just put some petri dishes on a plate and you zap it with lightning. And you're like, there it is. And that's not how it goes. So yeah, so she kind of lays all this out, and then she makes a shift into the part two, which my guess is probably the part that is required a lot of shift in her thinking, which she basically says, because again, she approaches this from a very narrative style of saying, you know, the first part of the book was the science, and then the second part of the book is how is this science going to be applied, and what should we think about how to control it or guide it or regulate it, so forth. And so I think she also does this very wisely, because she starts out talking about the chapter 5, which is the first chapter in the second part. It's called the Crispr menagerie. And she starts there mostly, if I remember right, talking about animals and plants. So we don't, the fifth chapter is basically discussing, okay, well, how is this going to potentially be used in agriculture? How is this potentially going to be used in things like, she talks about gene drives, which are ways of trying to control populations of vectors that can carry disease-causing organisms. So the great example of that is mosquitoes, which are an incredible disease vector for things from malaria to African sleeping sickness is the tsetse fly, but it's another insect. So I thought this was good, and I think she's done an excellent job here kind of trying to, this is, to me, so I'm actually married to a social scientist, I'm married to an anthropologist, so I kind of consider myself a little bit anthropology adjacent, and it's very interesting to talk to him about his research, because one of the things I like about microbiology is like you do an assay, and like the assay tells you this, or the assay tells you that, and it's not like you can have a large convocation of people interpreting the assay like in various different ways, like, well, in this country they would think this about the assay. You're like, that's not going to happen. It says this or it says this. But when you get into things like, you know, and I think maybe Nicole mentioned this before, the groundwork for how do you apply this technology gets back to things like, well, how does the USDA, the United States Department of Agriculture, define genetically modified foods versus how does the European Union define genetically modified foods versus how does India define genetically modified foods. So now you're into public policy, and which, for better or worse, is shaped by a lot of stakeholders who may or may not be perfectly well informed about the actual technology we were discussing. And, you know, as anybody who's got any training in evolutionary biology should do, she kind of harkens back to the fact that we have been doing a lot of genetic manipulation of domesticated crops and animals for a really, really, really long time. And we have a lot of breeds that are commonly consumed by people that are nothing like their ancestral relatives and that have been wildly changed by human intervention. And so she kind of comes down on the side of saying, like, look, we already do a lot of genetic modification. This is an accelerant, but it is not introducing anything new, because these are the same types of modifications that we would be trying to make with breeding. We're just speeding things up. Do you guys have thoughts on that? I know that genetically modified foods can be kind of a contentious issue, so I don't necessarily want to out anybody if anybody's got opinions on GMO stuff. But she was smart to kind of discuss that, because that's one of the applications of this technology, and I think it's something we're going to have to continue.
So I think she's very honest about her position on GMO, which I mean is mostly, it's permissive for her. And I think she tries very hard to clear up that, you know, because the whole point of CRISPR is that eventually, not now, but eventually, it's going to probably be a very high frequency on target source of editing a genome. That once you actually have the risk management from making your modifications as deliberately as possible, which really isn't addressed in the menagerie chapter, but later, that you really shouldn't be creating unanticipated downstream toxicities or effects in these modified crop plants. And I have to say, not very scientifically, I did enjoy the illustration of the very muscular animals, including the rabbit, which I was secretly hoping that they would have described as the jacked rabbit because it was a little bulkier, but they didn't go there. So that's too bad. I think she gave some very good examples in terms of the pest resistance, thinking about food security, thinking about a planet under climate change now, and the fact that not everyone's on board with the idea of engineering our way out of a crisis. But she's putting out some options there, which are quite compelling. And also the fact that she's layering some ethical issues that are already on top of ones that the public in general has already gone on board with. So the idea of editing a cow for hornlessness, so that even though it's destined to end up on our plates, there's one other event in the life of that livestock that is a harm-inducing event that is now subtracted from the lifespan of that animal. So we have to balance, as a society, what our value systems are. And I think this book does an important part of acknowledging that, but also bringing some potential clarification to the science, which is why I think this is such a great book to have out for the general readership. So that's what I'm going to say about the menagerie chapter.
I think probably this was one of my favorite chapters, just because of all of the examples, and also definitely because of the illustration of the jacked rabbit and the really muscular dog and stuff. That was actually something I didn't know about. Was a thing that you get the really, really muscular animals by just changing this one gene. I enjoyed reading about all these different examples and all the different applications it can have, and I was actually very excited of all the food-related ones because that could have really good uses for, like you said, climate change and food security. And I've always been very disheartened by the hate on GMOs because there's some very, very, useful ways that we could, world hunger, that kind of thing, that could be, resolved this way. And so I think one of the points that she makes in the book, I actually bookmarked it, that was where she basically asks, if you're using the CRISPR system and you're still ending up with a genome that looks essentially like a natural genome, if you're looking at GMOs, should you only consider the final product, or should you consider how you got there? And that was one of the questions that I thought was really interesting, was if you can end up with corn or something that is resistant to this disease, but we got that resistance gene from another species of corn, then why is that not okay, or why is that okay compared to adding resistance gene from a bacteria or something? And so I thought that was a lot about what this chapter was, and a lot of really interesting things that they can change with that. And the gene drives, like you said, we could get rid of, well, maybe not completely rid of, or maybe yes, mosquitoes, right? Because mosquitoes are a huge, huge problem for more than a handful of diseases. And I've been to areas where you can get yellow fever and malaria and all that, and it's very scary. Imagine people that live there all the time, and can you imagine having DEET covering your body 24/7? Because that could get really annoying. So that was one of the things that I really liked about this book. But obviously there's a lot of challenges in policy and how we bring the public into this, that I thought this would be a good starting point.
Yeah, I completely agree. I think if you were thinking from my perspective as somebody who's potentially going to teach this in classes, I think if you didn't want to make students or have students read the whole book, you could kind of bring some of the stuff from the beginning so they get a good grounding, and then this chapter on its own would just, you could springboard off into all kinds of questions about, and the title of the book, or the subtitle of the book, Gene Editing and the Unthinkable Power to Control Evolution, that starts to come to play, and it continues through the rest of this part, but it starts to come into play here, because just like Lisa mentioned, one of the things that's discussed is using something like a gene drive, we could potentially eradicate mosquitoes should we do it. That's, we've done that. We've eradicated we've eradicated a pathogen in the form of, we've got smallpox eradicated. So those are goals that we've actively had before. You can have a debate about that because basically you don't know if the virus is living or not, but we've caused the extinction of other living organisms that are no longer on the planet. But this would be an extremely deliberate and extremely if you eradicated mosquitoes with a gene drive based off of CRISPR, that would be a very, very deliberate, very, very particularly deployed thing to do that is going to change ecosystems potentially. Now, there's some quotes in here from people who are like, yeah, you just get rid of mosquitoes, that's fine. No one would care. But then you start to get back into things like, do you want to bring back the woolly mammoth? Why would you bring back the woolly mammoth? What's the woolly mammoth going to do? It's going to use Facebook and I don't know. But you ask questions like, when do you deploy this in a way that you're kind of like, why are we doing this? Like, why are we trying to kind of de-extinct some stuff? What's going to happen if we do that? So that kind of, and these are not new conversations because of CRISPR. They've been going on since somebody's done cloning, for instance, since we had Dolly the sheep, and you're kind of like, what's happening with this? So these are not new conversations, but I think CRISPR, the arrival of CRISPR has accelerated the urgency with which people feel like we've got to kind of talk this through. And then of course, so if we move on to the next chapters, so the next kind of, I think 2 chapters, perhaps, but she then transitions from plants and animals, and she transitions into humans very specifically. So the idea of CRISPR being deployed as a therapy in medicine. And here she distinguishes between somatic cell therapy, which is essentially let's say, for instance, you have someone with Duchenne muscular dystrophy, or someone with cystic fibrosis, or someone with Huntington's disease. The idea here would be that you could deploy CRISPR in a way to correct the mutation in those people's cells so that they themselves are cured. However, unless you go into the germline, those folks could still potentially pass on the mutated gene to their offspring. Germline therapy is basically saying, we're going to eliminate this from your lineage. You will no longer pass on the mutant form of this gene to your children. And she makes a couple of very important points, one of which is that we can't get ahead of ourselves too much because CRISPR is going to be a potentially useful tool for things that are single gene monogenic diseases where we can clearly, like cystic fibrosis, it's an ion channel, you can point to that ion channel, it's got a mutation in it, you're like, yep, that's your problem, we can fix that. But then you get into things like autism and cancer and heart disease, and if anybody knows anything about genome-wide association studies, man, take your pick. You just get like a whole range of like, these are all the scatterings of genes that have been implicated in this particular condition. We don't know, So you can't yet kind of deploy CRISPR in those contexts because we don't yet understand what the underlying genetic condition is. And always, with a lot of these, it's genes and environment together that are going to dictate your risk for some of these diseases. Apart from just the science behind it, there's also the question of, and she's very, I don't know if it's this chapter or later, but I appreciated that she touched on this. There's the idea of, so advocates for people who have particular conditions, either people who have that condition themselves or people who are caretakers for those folks, are often enter into these conversations to give the perspective of saying, you keep implying that you have to fix these people. And they present the perspective of saying there's nothing wrong with, for instance, oftentimes autism is an example that's brought up with this. So people who live with autism, some folks themselves or people who care for them are very vocal about saying, you're implying that there's something wrong with people with autism simply because they don't fit perfectly into the way that society has decided it's going to run things. Doesn't mean they're wrong. Doesn't mean they need to be fixed. It's just that they're different. And I think she touches on this. And I think that's one of the thorny questions that needs to get addressed as you start to decide, when are you going to use a technology like this? What kinds of conditions are you going to attempt to treat? There's a lot there, so I'll stop and see if that's kind of a broad. So I'm just curious what hit you guys about when she started talking about using CRISPR in humans. What kind of stuck out to you as being the most pressing topics?
So I'll go. I need like a few more weeks to possibly the rest of my life to think about this chapter, because I actually, on some level, I still want to give it a hard pass, but recognizing that it's also unfair because Dr. Doudna is not a policy expert. She's made a lot of inroads to try and figure out where she fits into that discussion. And I think the main, I guess, oversight, but it really doesn't fit into this book, is the idea that she does address that for those who are already born with a genetic condition, or for those who are advocating because those are lives that are inherently valuable, that they don't want sort of a pressure to, you know, convince people that they should make reproductive decisions a certain way, addresses one part of the other. I mean, she does mention that probably the eventual trajectory and also the bottlenecks of cost of genetic or gene editing for disease will mean that there's probably a socioeconomic tiering of who has access to corrective technology before probably large subpopulations of people whose lives will have disability and, you know, persisting effects of genetic disease. There is the long view of gene editing, which is that we can correct this early and have a healthier population. But there's the long view that is sort of not really addressed in the book, of public and government spending for infrastructures to make societies concretely accessible for everybody across a spectrum of healths and abilities. Because even if you do manage to get rid of all the genetic diseases, we're still going to grow old and die. So disability it's not going away. And I think there's a very big risk with talking about, you know, widespread implementation of gene editing to alleviate suffering when there's things like mandatory wheelchair ramps or relaxing on straw bans, or just, you know, less biologically intricate things that we do to make society as broadly as possible a place that people can live in. And it's a peripheral discussion to the one in the book, but it's one that definitely fits under the umbrella of what are we doing for everyone if there's this paradigm shift about how we address, you know, these potential genetic disorders. So I'm going to leave it at that.
That's definitely one of the thornier issues with the technology is well, I mean, this might be next chapter. She brings up like eugenics and how this could be like a huge problem, but also, yeah, like if you're going to make this a way of curing diseases and that kind of thing, then it's important that it's accessible to everybody and not just the rich. And there's just like, yeah, like you said, there's a whole discussion to be had on all of this. I think one of the things that, at least in my opinion, that this is her big question, right? Is do you just use it to change somatic cells, or do you use it to change the germline? And I'm sure that there's some diseases that I imagine most people would not oppose getting rid of. You know, things where like cystic fibrosis, for example. You know, it's a terrible condition, and you die very young, and you suffer a lot, right? So maybe stuff like that, but then, you know, where do you draw the line and the other things? And I think that is where a big part of the discussion needs to be. I suppose with the public, with people who study bioethics, who this is the thing that they do, but that is it's a complicated question, I think. But I mean, that we have the technology, I think, assuming we can get it to the point where it's safe and it's accessible, and then it would be very nice to, at the very least, use it in somatic cells for stuff like cystic fibrosis. And if we can never figure it out, cancer. That kind of thing. I think that was my thing. I think that this was also a bit more chapter 5 than chapter 6, but she also talks about engineering pigs for organ transplantation, and which I thought was also a very interesting potential way of using it. And I'm sure there's ethics on whether or not we should be using pigs for that, but I'm not an expert. But I thought that was very potentially interesting. And also, I forget what disease this was, but where you could potentially take somebody's bone marrow out and do gene editing on that and then reinsert it. And that a bone marrow donation is so difficult to begin with, right? So that being able to do that would potentially be really nice. But yeah, there's a lot of thorny questions starting even with the previous chapters, but I think it's especially heavy in these chapters, and that's very much where I start to get out of my depth. And
yeah, and I, so kind of to follow up on that, and I'm conscious of the time, I don't want to keep people too much longer. She kind of closes out by there's a chapter called The Reckoning, which discusses the meetings that she was a fundamental part of calling for, convening these meetings amongst multiple people who have expertise in the area, expertise in different areas, people who would intersect with this because of their role in public policy, patient advocates, journalists. And she calls back to the similar types of meetings that happened when recombinant DNA technology in the 1970s, when it became clear that you could put a gene from just about anything into a plasmid, into E. coli, and then you can start expressing things where they never were before, and stitching things together to take this domain from this protein, this domain from this protein, and smash it together to see what happens. Very similar ideas of should we be doing this? How should we be doing this? What are the implications? Kind of came to the front with that technology. So she very consciously calls back to that, and then prompts these meetings. And the way she kind of leaves it is to discuss how those meetings ran, and then also she closes out kind of with her current, at the time of her writing, her current thinking on what does she think it would be appropriate to deploy CRISPR to do in agriculture and in medicine and overall. And I think one of the things that we've kept coming back to, and as somebody who went to a liberal arts college and teaches at a liberal arts college, this is where I'm going to plug the liberal arts. Yes, come to that one. But this honestly is where you have to have people who have spent their time studying ethics. And you have to have people who have spent their time kind of thinking about policy. Because this technology might have been developed in a research lab where you're not constrained by society, but it's going to get deployed in the society that we have now, with all those constraints and all those limitations and all those opportunities, but it has to intersect with society as it is. And sometimes I think we're scientists, and we kind of hope that we're headed to a Star Trek future where everybody's equal and there's no income and everybody just gets their hot Earl Grey tea from the replicator and everything's great. But that's not how this technology is going to be deployed. That society is very far from us at this point. And so we've got what we've got with all the inequalities and so forth that are incumbent with that society. And so we have to think very carefully about how this is going to intersect with lots of different things, with access to resources, with social inequality, with financial income inequality, all of this. And so I appreciate very much that she stepped outside of the lab and said, we've got to bring people in who know this stuff. We've got to bring patient advocates in. We've got to bring in people who understand health inequality, who studied how the American health system works versus the European health system versus the health system in China, because my gosh, that's going to impact who gets access to what technology. And she's kind of left, especially not to get political, but especially given our current situation in the world where you turn on the news and everything is just a burning dumpster fire of horror. She ends with a very hopeful sense of saying, like, you know, I think we can get to a point that we can treat this in a conscious and in a conscious way, that we are thinking very clearly about what it is, what it offers, and what we need to be cautious about. And I'd like to think that too. I'd like to think that we can do that as well. But it certainly is going to take a lot of effort from a lot of different facets of society and a lot of different people to kind of figure out how to guide this, which is part of the reason why I think she wrote this, because I think it comes through very strongly that she feels like people like my mom, who, you know, sent me, I've just read an article, I'm probably gonna send her this book now. Now she's done it. Now she's gonna have to read this book. But I think that part of the reason she wrote this book was for people who hear the term or see an article and then kind of want to know what's going on with this. You know, when am I going to start to see this, you know, in agriculture or in my doctor's office, and to bring people into that conversation, which I think is a really excellent reason to write a book like this. Do you guys have closing thoughts along those lines?
I'll go. So this is probably one of the more optimistic things I've read. I spend a little too much time on Twitter, so this definitely is one of the more optimistic things I've read recently. And I'm just going to rehash on the clarity. I love this book, but I also think it's a narrative about something that, you know, scientists kind of take for granted that they control, because we've been manipulating genetic material and sort of selectively exploiting some aspects of genome dynamics for a while. Not a long while, but a while. Crispr is definitely on the shorter part of a while, but I mean, these ideas that we are in societies where we have certain elements of control, but also the idea of control in terms of disease prevention, the idea of control in terms of having health or agricultural or food security issues advocated for and addressed. I mean, that's a criteria for feeling included in your society. And I think that's maybe the way we think about these as a group and as a combination of policy stakeholders, which of course includes the public, is how we think about when and where we are going to deploy this. And one of the things that's coming to my mind right now is at the university, I was at UNBC, there is a biobanking initiative that's targeting Northern and Indigenous community health needs, which definitely, when you do conventional epidemiology and think about a frequentist approach to defining disease, this is not, you know, a percentage. If you were going to make decisions on that, that would be like a high and dominant one. But it is in a community that's been traditionally not well described and not well screened for medical conditions that are important to them. So if you have that happy intersection of focusing on pathologies that are actually very easy to treat with CRISPR or gene editing, and creating an opportunity to actually address, you know, something in a population that never gets the advocacy and the attention to make their section of society better. I think those are where these pieces of seeing the really good applications where everyone is invested and everyone is on board to gain. So is this a just application of the technology? Is it one that people can live with? Is it as equitable as possible? And we're never going to hit those at 100% every time, but I think those are sort of the values that have to draw us together in figuring out what we're using this for.
I really, really liked your points. I thought that was very good. I think you guys have said most of it, right? Like she ends on a hopeful note, and the hopefulness that we can use this in underrepresented communities and that kind of a thing, I think is a very, very hopeful outlook for the future. And it's kind of how I tried to read this book, is like, yes, we can probably use this for good. So what I actually might do is, at least part 2, is anybody that I know in my lab or that I've talked to, I want to at least make them read part 2 so that they can start to think about like the implications of the things that we do. And especially if I have friends that work on CRISPR, I think this is a good book for not just the public to read so that they understand that this technology now exists and can join the discussion, but I think it's an important book for scientists to read so that we start to remember that we do these things, but also society is still here and we need to think about how it affects them.
Yeah, I think that's an excellent point. I think it's written for, it's definitely suitable for both a non-scientist and a scientist audience. I think you're absolutely right. And having said that, as a scientist, I'm really glad I read it. I thought it was a great book. It was really well written. I thought it was kind of a swift read. There were a couple of things that I did have to visit back to, but I don't think you're going to find yourself. If you're the type of person like my husband who basically reads 5 pages before he goes to bed, I don't think you're going to find yourself like, oh my God, like I think you'll make it through the book. So I definitely encourage people to read it. So with that, I just want to thank you guys for joining me for this. Lucy is new. This is her very first time joining us, and Nicole is basically taking up all the hotel Wi-Fi in Montreal to try to get here. So I really appreciate that. Our next Micro JC Book Club discussion will be in August, and I don't remember the date off the top of my head, but it's always announced on Twitter if you look at #MicroJC. If you'd like to join us for that, we would definitely be glad to have anybody join us. We're always happy for new and returning people. And we'll be talking about, we'll be discussing a book called, August 9th. Thank you. Lucy just pointed out it's August 9th. I completely forgot. So August 9th at 1 p.m. Eastern, we'll be talking about Improbable Destinies, which I haven't started yet, but I'm very excited about because it's a bit more of, if you're an evolutionary biologist, I think it'll be up your alley. And if you're not, this is probably going to be a great introduction to kind of thinking about chance and contingency and how populations evolve. And so more than thinking about how to direct evolution, it's more about how evolution kind of shapes populations and how that works. So I'm really excited about that. So with that, I will close out our broadcast, and thanks for everybody for being here today.